Okay. So last time when we were discussing, I think we ended on these mechanisms for test transduction. So basically, the topic that you're looking at right now, for those who are joining us for the first time, is known as gas station or test physiology or physiology of test. So where we are right now, we are looking at the mechanisms of test. So as you can see, this is just a simple flowchart, which can be able to help you during an exam, whereby if you have forgotten very simple concepts, you can be able to look at this diagram and be able to capture one or two things and be able to recall the data or the information that you have. All right, so now we are looking at the mechanism of test transduction. Okay? So like we have got about 5 basic tests, okay? So like we discussed in our previous video, there's bitter, sweet, umami, sour, and salty, right? So now, uh, let's start with bitter. So bitter test, okay? So remember in the previous session, we talked about what bitter consists of. So basically, these are just chemicals, right? That are going to be dissolved in saliva, and then they're going to be picked up by the right receptors on the test buds, okay? Yeah. Remember, the ones which are sensitive on the test bud is cell number 3 or type 3 cells. So these are the chemicals that consist of a beta test, depending on what you're consuming. So now, it binds the G protein coupled membrane receptor. Remember, receptors are the one that detect the change in an environment or a stimulus. So we are going to have IP3, inositol triphosphate, and calcium levels are going to be elevated, which is going to cause the opening of the therapy channels, which is going to cause what we call depolarization. When you hear the word depolarization, means an action potential has been initiated, and there has been an influx of a certain type of chemical or ion going into the cell, hence causing the inside of the cell to become more positive, while the outside of the cell becoming more negative. That means an action potential has been generated. So basically, when we started this topic, remember we talked about something that was very important, which was we are trying to communicate something, right? And how are we going to do this? We are going to convert this chemical message, which houses a beta tester, this chemical message, into an electrical signal. So from chemical to electrical signal, because or electrical action or the action potentials that are going to be picked up or interpreted by the brain. Remember, we talked about first order, second, and third order neurons. Okay, so that passage or that pathway involves the communication between the tongue and the test centers in the brain. So that's what it involves. So the brain can only detect action potentials, right, as the true messages. So that's what we are looking at right now. That's what we mean by transduction mechanism. You're trying to convert from chemical to electrical. So this is what is happening. Secondly, you look at this. This is what we call sweet umami. All right. Yeah. So this is another type of test sensation. There's sweet and there's umami. So these chemicals that are detected by the specific receptors on the test buds, okay, they bind to a G protein carbon membrane receptor, which is able to sense what type of chemical is that. Once it picks up those chemicals, it's going to cause the same thing that happened under beta. IP3 elevated, calcium elevated, opens therapy channels, which also cause what we call depolarization, meaning an action potential has been initiated. Then when you look at this, you're going to have what we call sour. So sour taste, here we are privileged enough to have showed even the type of chemical that is involved. For those that don't know, it's known as hydrogen, okay? Ions. So these are the ions, or the protons as they call them. They're going to be picked up, or they're the ones that cause a change in an environment. They are the one that cause a stimulus. So enters through the membrane channels, enters through the membrane sodium channels. So there's what we call NAC, okay? So once it happens like that, it's going to cause what we call depolarization, meaning positive on the inside, negative on the outside. Operation has what? Kicked off. Then when you go to the salty, sodium channels are the major, the sodium ions are the major as you can see. Enters through the same membrane sodium channels in NAC. Then after that, you're going to find that there's going to be what we call depolarization. So this is just a summary showing you how these things are happening. It's a summary. Okay, no hard feelings. So now let's go to something that is a bit more. Let's go to something that is a bit more, yeah, maybe a bit more detailed. So now, however, several other receptors are also involved in taste sensation. The transduction mechanism is different in each taste receptor. Okay, so however, several other receptors are also involved in taste sensation. Transduction mechanism is different in each taste receptor. That's what they talk about. Now, sweet receptor. Receptor for sweet taste is the G protein couple receptor, like we looked at in the previous. So that's what they mean. Okay, binds to G protein coupled membrane receptor. That's what it means. CPRC, GPCR. So the sweet substance binds to receptor and causes what we call depolarization via what we call the cyclic MP. Okay, then the salt receptor for salt taste is called epithelial sodium channel INAC. Epithelium sodium channel. It acts like an epithelium sodium channel receptor in other parts of the body when sodium enters. So it's just the same thing. What you learned under cell physiology, it's the same application. It acts like an INAC receptor in other parts of the body. When sodium enters, the receptor releases what we call glutamate, which causes what we call depolarization. Okay, so when it enters, it causes sodium to enter. This releases glutamate, which causes the cell to do what? Undergo depolarization. Sour receptor. Sour sensation also has the same INAC receptor. The proton or hydrogen enters the receptor and causes depolarization. It's believed that beside INAC, other receptors such as the such as the hyperpolarization activated cyclic nucleated gated cation channel, HCN, also are involved in sour sensation. Beta receptor is a GPCR. In beta receptor, the sour substance activates phospholipase C through the G proteins. It causes production of inositol triphosphate IP3, which initiates polarization by releasing calcium ions. So guys, I want you to go in your own free time, read this concept under guide on, try to understand what's happening here and appreciate the information. Umami receptor is called metabotropic glutamate receptor, okay? mGlu-4. Glutamate causes depolarization of this receptor. Exact mechanism of depolarization is not clearly understood. Activation of umami, as you know, umami was recently discovered in Japan. Activation of umami test receptor is intensified by the presence of guanosine monophosphate, GMP, and inosine monophosphate, or IMP. So this diagram is just trying to show the mechanisms that happen there. And the salt, sour, sweet, bitter, and umami. Okay, so these are the different types of channel proteins that are there, or signaling molecules which are there. So you can go through this diagram. This is just what I'm talking about. So this is just a summary showing the stuff we are from looking at. Okay, so now applied physiology and abnormalities of taste sensation. So basically, when you look at these diagrams, you're going to find that it's easier for you to understand the pathophysiology if you have already understood the normal physiology, okay? It's easier for you to do that. So, abnormalities of taste sensation. Algesia. Algesia, it refers to the absence of taste sensation, okay? Yeah, so it's one of the culprits that is present in physiology of taste, or abnormalities that come about under physiology of taste. So algesia, it refers to the absence of taste sensation. Algesia of, causes of algesia are lesion of the medulla, so lesions of the mandibular is one of those. Lesion of facial nerve, okay? So lesion of the mandibular, diversion of the trigeminal nerve through lingual branch, of which the caudotepinae nerve reaches the tongue, cause loss of taste sensation in the anterior part third of the tongue, meaning you lose sensation of taste. Lesion of the facial nerve,
also leads to the loss of taste sensation in the anterior two-thirds of the tongue. Lesions of the glossopharyngeal nerve are associated with the absence of taste sensation from the posterior third of tongue. Drugs like cartopori and isoalumin, which contains groups causes temporary loss of taste sensation. So these are just talking about the different types of taste sensation, which can you be able to do in your own free time. And this is the end of this short video containing